Welcome to the Human-Animal Interaction Lab at OSU's instructional video on the collection and extraction of canine DNA. The product we use for this collection is called the Origene swab and it's produced by DNA DNA. This product has been replaced by a similar product known as Performagene. However, the procedures are very similar. First, we'll start out with a saliva collection. The collection must be done with the animal having been fasted for a minimum of 2 hours to avoid contamination. The upper and lower lips are extracted from the side of the teeth, and the swab is placed on the lower part between the lower molars and the cheek. This is an area where saliva will typically pool. The swab should be left there for a minimum of 30 seconds and up to 60 seconds. It can be moved side to side, avoiding touching the outside part of the lip. It's also important that the animal does not chew on the swab. The present slide has a list of the required equipment and reagents. In addition, it will be necessary to have the proprietary purification solution, which is provided by DNA DNA, and a vortex, as well as a bucket of ice. Visible is our sample in the incubator at approximately 50 degrees centigrade. In step 5, 250 microliters of the saliva sample will be transferred to a 1.5 milliliter microcentrifuge tube. To the 250 microliters of the sample, we are going to add 10 microliters of the purification solution, which is provided by DNA DNA. After addition, the solution needs to be vortexed for 5 seconds. It's also important when placing the tubes in the microcentrifuge to have them placed in a known orientation. We typically put the hinges to the outside. This will be important later. The top is put on the microcentrifuge and the centrifuge is started for 5 minutes. After the centrifugation, we are going to extract the supernatant without disturbing the pellet, which you will see visible in a moment. This is the reason for keeping the typical orientation. Because we know the orientation, the pellet will be in the bottom on the upper side, as shown here. We can use this to help us extract the supernatant without disturbing the pellet. To the supernatant, we're going to add 15 microliters of a 5 molar solution of sodium chloride. To this solution, we are going to add 300 microliters of a room temperature 95 to 100% ethanol. After the addition of the ethanol, we will invert the tube gently for 10 times. After having been inverted, we're going to leave the sample at room temperature for approximately 10 minutes. Then place it again in the microcentrifuge in again a known orientation. This time we will centrifuge it for 2 minutes. After the second centrifugation, we are now going to extract the supernatant, but in this situation we will keep the pellet and discard the supernatant. Again, it's extremely important to keep the pellet as calm as possible and not have it mix with the solution above it, the supernatant. Knowing the orientation of the tubes will help with us to identify the placement of the pellet in the tube. After extracting the supernatant, we are going to add 250 milliliters of a 70% ethanol. This should be done gently as to not disturb the pellet. After adding the ethanol, the tube should be placed in a quiet place for 1 minute. After 1 minute, the ethanol will be extracted, always being careful not to disturb the pellet. The orientation again will help us to identify the placement of the pellet within the tube when we're extracting the ethanol. It's often very difficult to see this, so this will help us to identify where the pellet is. After we extracted the ethanol, we are going to add an 8 molar TE solution. This is for the rehydration of the DNA. After our addition of the TE solution, we'll again need to vortex for a minimum of 5 seconds. After our sample has been reconstituted, it can be kept at room temperature for approximately 3 months or frozen indefinitely. The final procedure will be taking our sample and quantifying the concentration of DNA in the sample. This is done using a photospectrometer, in this case a nanodrop. The nanodrop is prepared by cleaning the pedestal with deionized water, and then 2 microliters of our solution, of our sample, is placed on the pedestal. The information gained from this procedure will allow us to know the concentration of DNA in our sample and the relative purity.